I think we have quite a few people already, so maybe we can get started.
Yes.
Yeah. Okay. So welcome everyone to today's Theory of Living Matter seminar series. And today we have Jakub Sedzinski, who's an associate professor at the University of Copenhagen. So he got really interested in tissue mechanics during his PhD, where he started working on his PhD with Eva Paluk when she was at CBG Dresden. And then he slowly moved to Texas, where he worked with John Wallingford and got interested in frog development and morphogenesis and the role of mechanics. And today he's going to talk to us about the mechanics of basal body patterning during multiciliated cell morphogenesis. So
Correct.
all yours now.
Thanks a lot. So thank you very much for the invitation. It's really a great pleasure to be part of this seminar series. So yeah, just before I start, just yeah, feel free to interrupt anytime if something is unclear, so that we can kind of figure these things out. So indeed, I would like to talk about this project, which is about mechanics of basal bodies. So this is very much unpublished work from the lab, and would be also great to get your feedback and suggestions. So please be very critical about the work. But before I start talking about this basal body dynamics, let me kind of put it into the perspective what we are doing in the lab. So in our lab, we are very much interested in understanding how to build specialized tissues. So tissues having well-defined cell type composition and complex 3D architecture. And we want to understand how to build such a specialized tissue starting from unspecialized cells, something like stem cells. And we want to understand how is this transition from unspecialized cells to specialized tissues orchestrated, both in space and time. But we also want to understand how the lack of this coordination, lack of this orchestration actually leads to the disease states and formation of the tissues which having a wrong cell type composition and a wrong architecture. And we are addressing these questions in a context of mucociliary epithelium. And mucociliary epithelium is a specialized tissue lining respiratory tract. So this tissue functions in a following way. So here you can see a section through trachea. This is actually a rodent trachea, but very much the same thing is happening in human. So what do you see in here is the flow of the substance called mucus. And mucus is a very viscous liquid that traps bacteria, dust particles, pretty much everything what we breathe in. And this mucus is generated by specialized cell type called goblet cells. So these pink guys. So now, in addition, we have also ionocytes, and actually ionocytes regulate the viscosity of the mucus by controlling the ion composition across the membrane. And additionally, we have also these funny looking cells called multiciliated cells. And these multiciliated cells, they have these long protrusions called cilia, which are sticking from the apical side of the cell. You can maybe see in the background, actually these cilia are beating in a synchronized way. And by doing so, they actually move mucus from the left to the right. So what else we have in here? We have also club cells, and club cells actually produce anti-inflammatory peptides. And finally, we have basal stem cells. So basal stem cells, in principle, they can regenerate all of these cells upon injury. So these cells together, they are working perfectly in a perfect synchrony. So they perfectly synchronize, forming the first line of defense against all of these pathogens and dust particles. However, this protective function of the mucociliary epithelium is heavily impaired in patients suffering of chronic respiratory diseases, for example, in asthma or chronic obstructive pulmonary disease. So in these patients, this mucociliary epithelium is heavily remodeled, and it's remodeled on multiple levels. So for example, we have in these patients, they have changes into the cell type composition. For example, in the COPD patients, we have increase in number of the mucus-producing goblet cells, and this is a condition called goblet cell hyperplasia. We have also changes as to the overall tissue morphology and the shape of the cells. So often cells undergo epithelial to mesenchymal transition. And finally, we have also changes to the mechanical properties of the tissue. For example, if you have asthma and you have this bronchoconstriction, then the mucociliary epithelium is actually compressed. And this is basically leading to changes in the mechanical or rheological properties of tissues. So now if you think about it, so if you think about both development and this pathological remodeling, both of these processes are actually driven by three dynamic or emerging properties of tissues, namely cell fate, cell shape and movement, and tissue mechanics. So cell fate is driven predominantly at the level of gene expression. So these genes actually drive the progression from unspecialized cells until the full maturation, so until the full identity of the cell, through the sequence of these steps, which are called cell states. So what is interesting that as cells are changing their fate by regulating gene expression, they're also changing their shape. And in that case, the shape and the movement of the cells is actually regulated by actomyosin forces, which are generated by the cortex of the cell. And now these forces actually propagate across the entire tissue, forming the base for tissue mechanics. And tissue mechanics defines how actually the tissues actually respond to external forces, for example, stretch or compression. So now in our lab, we're actually trying to integrate all of these three parameters together. So cell fate, cell shape and movement, and tissue mechanics to basically understand how these parameters working together during the formation of the tissues during development, but also during the pathological remodeling. So we started looking at the interplay between these processes, and looking at the differentiation of multiciliated cells. So again, what we know about multiciliated cells morphogenesis, based on our work and others, is that these specialized cells, which we here see with cilia with the number 4, they actually originate from basally positioned stem cells. And these stem cells, upon specification, they actually migrate from the inner towards the outer layer of the epithelium. And once they reach the superficial layer, they actually insert there and open the apical domain. And this is actually required to start growing cilia. So to study the processes related to the morphogenesis of multiciliated cells, we actually use a frog system of frog embryos. And the reason is very simple. So this is on the left-hand side, this is the monster we're working with, so Xenopus laevis. And on the right-hand side, we have actually the frog embryo. It's actually quite big, so 1 millimeter in diameter. And you can actually appreciate these blue spots. And each of these blue spots is actually a single multiciliated cell that you can appreciate better on this scanning electron microscopy picture. So you can see again, beautiful tuft of multiciliated cell, which are actually surrounded by goblet cells. And you can actually see the vesicles. So these are the mucous vesicles. So again, what we know is that these cells, as they move towards the superficial layer, they insert at the position called tricellular junction. So tricellular junction is a vertex which is built by 3 or higher number of cells. So again, upon insertion into this vertex, they open the apical domain and then they start growing cilia. So experimentally, this process looks pretty much like this. So here on the left, so in green, we see the expansion of the apical domain within the progenitor of multiciliated cell. And in magenta, we see actually the dynamics of actin in the surrounding goblet cells. So in this work, we showed that the dynamics of the differentiation of multiciliated cells actually depends on the force balance between the forces generated within the apical side of the progenitor cell and also forces generated within the surrounding tissue. But when it comes to the interplay between these three different parameters, there's another kind of level that we have to understand in here, which is not at the level of cell and tissue, but also at the subcellular level. So this is particularly important again when it comes to the multiciliated cell and the structures called basal bodies. So basal bodies are actually organelles which are nucleating cilia. So you can see here schematics of multiciliated cells. You can see this schematics of axonemes, which are, as you saw before, they're beating in a synchronized way. And these kind of spherical structures, they illustrate basal bodies. So these basal bodies are actually embedded within the apical domain, and they're embedded within the actin network here in pink. So what is very interesting that during differentiation of multiciliated cells, this structure, so these basal bodies, you can think about this again as a modified centrioles, they're actually amplified or generated within the volume of the cell, and actually they are generated kind of at the bottom part of the cell. So as a cell, a multiciliated cell undergoes differentiation, these structures, so basal bodies, they move from the bottom towards the apical side of the cell. They get distributed within the apical side and then get patterned. So this is very important because this proper organization of the basal bodies within the apical side of the apical domain, this is actually very important to get the synchronized beating of cilia. So what is another aspect that we have to consider here is that as basal bodies are basically moving towards the apical surface, the apical side of the cell is also expanding to basically accommodate the increasing number of the basal bodies. So we have to here basically two processes. On the one hand side, we have basically dynamics of basal bodies. On the other hand side, we have basically expansion of the apical domain. So to illustrate experimentally how it looks like, this is basically how the process looks like looking at the fluorescent images. So again, on the left-hand side, we see basal body dynamics looking at the site of the multiciliated cell progenitor. And on the right-hand side, we basically only see the expansion of the apical domain. And you see that, first of all, let's say if you look at the left-hand side, we see that these basal bodies are moving up. And then when you look at the right-hand side, we can see this very nice, basically spreading of basal bodies within the apical domain and the patterning of the basal bodies within the apical domain. So this is quite interesting, but in order to understand this process, we have to understand basically three parameters or three processes. So we have to understand how these basal bodies basically move apically, and also how they get spread within the apical side, and finally how they get patterned. So we actually have no clue about none of these processes. So basically, first what we did in the lab, we started addressing the second and the third part of the process. So basically how basal bodies get distributed and patterned within the apex, so within the apical side of a cell. So here we have to consider two elements. So on the one hand side, we have to consider the dynamics of basal body itself. And the second player that we have to consider here is actually dynamics of actin network during the expansion of the apical domain. So here again, what I'm going to talk about in the next couple of slides is going to be how these basal bodies here marked in green are distributing and patterning during the expansion of the apical domain, which is basically built by actin network in pink in the schematics. All right, so what is also very interesting when we talk about the spreading and patterning of basal body, there's actually a very strong correlation between number of basal bodies and size of the apical domain. So for example, here, if we plot the number of basal bodies, so in short, BB, on the y-axis and in time, so within the expansion of the apical domain, so 150 minutes in that case, we see that as the apical domain, as the apical domain is expanding, the number of basal bodies is increasing. If you then plot, in that case, on the right-hand side, the number of basal bodies and against the surface area of the apical domain, you see very nice, almost linear correlation between these two parameters. What is even more interesting that, as I mentioned before, as apical domain is expanding and as basal bodies are inserting into the apical domain, the basal body get patterned. So this could be, quantified. This patterning could be quantified in a different way. So here we actually look at the nearest neighbor distance between the basal bodies. So you can see here that as as the apical domain is expanding in time, initially there's less patterning, and then as the new basal bodies are coming into the apical domain, we start seeing this very nice patterning evolution. And basically at the end, we have more or less about 1 micrometer distance between the basal bodies. So you can actually see how it looks like this process of patterning on the still images, which are here on top of this slide. All right. So the very first thing that we did to understand how we actually get the spreading of this basal body and this patterning, we wanted to understand how these basal bodies actually move within the expanding apical domain. So how they actually behave within this actin network. So to do so, we actually measure mean square displacement. So how these basal bodies are moving within the actin network. So here, just to remind you, by basically quantifying mean square displacement of basal bodies within the actin network, we can determine whether basal bodies are diffusing. So if the slope of the mean square displacement is 1, then we know that we can approximate that the basal bodies are diffusing. If the slope is higher than 1, then basically this suggests that the basal bodies are actively moved by actin network. And if the slope is lower than 1, this would suggest that the basal bodies are in fact constrained within the actin network. So specifically what we did in here, we imaged the movement of basal bodies within the apical domain with a very high temporal resolution. So this is about 21 milliseconds. So what we did in here, we basically take apical domains of different sizes. So for example, I know here, let's say 100 micrometers square, and image this apical domain with the 21 millisecond resolution for 10 minutes. And then we find another apical domain, which is of different size, and basically did the same. So image for 10 minutes with the 21 millisecond resolution. So this is how the mean square displacement looks like. So for example, we see here that if we start looking, for example, at the mean square displacement, looking at the long time scale. So here, I don't know if you see my cursor, and look at the slope and plot this slope for hundreds of experiments we did against the surface area. This will look like this. So again, on the y-axis we have alpha value, which is again the slope of the mean square displacement, telling us whether the basal bodies are diffusing, actively moved, or subdiffused. And on the x-axis we have actually the surface area. So given the variance of this data, we can say that largely basal bodies are in the diffusive regime when it comes to these large time scales. However, there is some sort of tendency that towards the bigger sizes of the apical domain, they are somehow in the subdiffusive regime. And I'm going to kind of explain this a little bit more in details in the next slides. So this, as I said, kind of suggests that initially the basal bodies are more kind of diffusing, and then towards the larger sizes of the apical domain, they're actually getting more constrained. And this is, in fact, very much in agreement what we visually see when we track this basal body. So, for example, if you look at the smaller size of the apical domain, so as we call it during expansion, then these tracks are actually longer. And when we look at the expanded apical domains, then these tracks look shorter. And actually, we can quantify this length, so-called end-to-end length. So basically the length of the entire trajectory, at the very early stages of expansion and during the expansion and towards the end of the expansion. And this is actually plotted in here. So again, here we can see that on the y-axis is the so-called end-to-end length. So how long is the trajectory of the basal bodies? On the x-axis, we have actually the surface area. And again, we see that initially the length of the trajectories are longer and then getting shorter towards the end of the expansion of the apical domain. All right. But now if we look at the mean square displacement, kind of looking towards the short time scales, we have actually a very different perspective or very different view on the movement of basal bodies. So here, what I illustrate is a mean square displacement in time. And you can see these three like plots of mean square displacement. So these are representing the mean square displacement for cells at different stages of expansion of the apical domain. So for example, red color, this is for the apical size of the 47 micrometers square. The black one is 140 and the blue one is 305. So what we see here that when we consider short time scales, the basal body behavior is actually subdiffusing, meaning constrained. But there's another interesting thing that we see in here. So actually we see that this transition from this subdiffusive regime to the diffusive regime is actually happening at different times. So for smaller sizes of apical domain, this transition is happening at the earlier stages. And for the largest sizes of the apical domain, this transition is happening at the later stages. So now what we did, we actually looked through all of these cells that we have, so hundreds of cells, and basically plot the transition time against the surface area. And this is the plot. So on the y-axis, again, we have transition time. So again, this is the transition time when the basal body transition or shifts from the subdiffusive regime towards the diffusive regime. And on the y-axis, sorry, on the x-axis, we have the surface area of the apical domain. So what we can see in here that initially for the smaller sizes of the apical domain, this transition is very fast. Let's call it around 5 seconds. But now, towards the later stages, towards the bigger sizes of the apical domain, the transition is longer, about 10 to 15 seconds. And what is also interesting, we see here some sort of shift or transition around 150 to 200 micrometers squared. So again, we increase this transition time, and then at some point we reach a plateau. So this dynamics of the transition time strongly suggests that there is some sort of reorganization, remodeling of actin network, because these basal bodies are positioned in the actin network, and we see that some sort of changes when it comes to the transition time, most probably related to the cross-linking of actin network. So with this in mind, we start looking at the organization of actin network within the expanding apical domain. So here we can see the image sequence of apical domains here visualized with actin at different sizes of the apical domain. So now so again, actin is in pink and basal bodies are in green. So now if we zoom into these regions, we see that when apical domain is small, let's say here for 70 micrometers squares, these basal bodies actually kind of close together. And towards the larger sizes of apical domain, in that case, for example, 212 micrometers square, we start seeing this more a regular pattern of basal bodies, and actually, in fact, we see this reorganization of actin around the basal bodies. You can think about it as some sort of pocket in which the basal bodies are embedded, in which the basal bodies are sitting. So what we decided to do, we wanted to actually quantify the reorganization of actin network, and to do so, we actually measure the intensity of actin at the position where basal body is present to the intensity of actin around the basal body. So again, at the basal body and around the basal body, so basically the donut structure. So now to quantify this kind of reorganization of actin network, so basically formation of actin ring around the basal bodies, we basically use the CDF, so cumulative distribution function, to basically quantify how much, or you can think about it, how easy is to find this actin ring around the basal body as we kind of move in time, or in that case, as we kind of expand the apical domain. So as you can see here, as we move within the size of the apical domain, this cumulative distribution function, they separate from each other, meaning that the cumulative distribution function that describes the intensity of actin at the position of basal bodies and at the position around the basal bodies, these two kind of lines or plots separate from each other, again telling us that you start building more and more actin around the basal bodies. So now, to understand this process, how this process looks for many cells, we actually can plot the shift between these two CDFs at the 50% of the value, and this plot is actually shown in here. So what we see in here, that as the apical domain is expanding, basically we start growing these actin pockets around the basal bodies. So you can also see it on the right-hand side. But what is very cool in here, that now if we plot together this plot together with the one that I showed you before, they actually show very much the same description of the situation. So again, on the left-hand side, we basically describe the organization of actin network around the basal bodies, looking at the organization of this actin meshwork around the basal bodies. And on the right-hand side, this is basically looking at this transition time when the basal body transitions from the subdiffusive to the diffusive regime. Yeah. So in fact, we are describing the behavior of the system from two different perspectives. So on the left-hand side, we describe how the actin network is reorganizing as the apical domain is expanding. And on the right-hand side, we basically describe how basal body dynamics is changing in time as the apical domain is expanding. But in fact, both of the kind of quantification, they basically say the same, that there is some sort of reorganization of actin network, and this actin network reorganization is somehow, has some sort of a shift point, which is happening about 150 to 200 micrometers square. So what does it mean? So looking at these results, we hypothesize that this probably means that as basal body reaches the actin within the apical domain, it starts getting cross-linked. So initially, in the very beginning, so when the apical domain is small, the cross-linking is very short-lived, meaning that, let's say, it takes few seconds until this cross-linking will turn over and the beta bodies can be kind of free. So basically, they initially get caught by actin network, and because of the turnover of actin network, then they can move from the subdiffusive to diffusive regime. But now, as apical domain is expanding, the cross-linking around the basal body is getting stronger and stronger, meaning that the turnover is also longer for the basal bodies to get released from these pockets. Yeah. So if this is true, if our hypothesis is true, then we expect that if we perturb now the actin reorganization, in that case, cross-linking of actin network, we would expect some sort of changes happening as to the organization of basal body within the apical domain. And this is actually what we did. So here we perturb or knock down alpha-actinin 1, which is the major cross-linker of actin within our system. And this is actually what we see. So on the left-hand side, we look at the signal of actin, and on the right-hand side, we look at the signal of basal bodies. So what we see in here, that first of all, the expansion is much slower compared to the contrast situation. But what is very interesting, what was very intriguing, is that actually basal body, instead of spreading and getting pattern within the apical domain, they're actually getting clumped. So this is, very much in contrast to what we know from the contrast situation. So again, just to remind you, on the top we see, the process of apical expansion and basal body patterning within the contrast situation, and at the bottom, I'm plotting here or visualizing the dynamics of apical actin and also distribution of basal bodies when we knock down the major cross-linker of actin. So in our case, alpha-actinin 1. All right, so okay, so what else we did in here? So seeing these changes into the distribution of basal bodies, we also basically quantify the mean square displacement for basal bodies in the knockdown condition. So basically, we did exactly the same experiment that we did in contrast. So basically, image the dynamics of basal bodies within the expanding apical domain with this 21 milliseconds time resolution over 10 minutes for different sizes of the apical domain. So just to remind you, on the left-hand side, this is this transition time, from the subdiffusive to diffusive regime for the control situation. And on the right-hand side, this is how this transition time looks for the situation where we knock down alpha-actinin 1. So the very first thing that you notice in here that this transition time is actually much faster when it comes to the alpha-actinin 1 knockdown. So this would suggest that there's not so much of the cross-linking happening within the knockdown condition as opposed to the control situation. All right, but what was actually quite interesting is that when we look at the basal body density, the basal body density is actually not changed. So meaning that even though the cross-linking is perturbed, the number of basal body inserting within the apical domain is actually not really changed, meaning that we still have this very nice correlation between the number of basal bodies and the size of the apical domain. And on top of that, the global density of basal bodies is actually not changed compared to the control situation. But what we observe is again this clumping, so that the basal bodies, even though the density is, let's say, normal, as it should be in a control situation, the basal bodies are clumping within the apical domain when it comes to the alpha-actinin 1 knockdown condition. So we want to understand, so what is actually the reason for this clumping upon knocking down alpha-actinin 1? So we want to understand a little bit better the difference, or actually what's the mechanism of the clamping of the basal body upon knocking down this crosslinker. So for this, we quantified the dynamics of basal body kind of insertion within the apical domain, basically trying to understand when in the process of expansion of the apical domain we start seeing this clamping. So if we, for example, look at the wild type situation, so here what we did specifically, we basically divided the apical domain into four parts, so quarters, and we basically look where the basal bodies are inserting within this quarter, and basically quantify or plot on the right hand side the density of basal body within the expanding apical domain. So let me just play it again. So again, here we just plotting the density of basal bodies within this four parts of the apical domain. So this is how it looks in the wild type situation. So in contrast, so what we see in here in the first place is that there is basically no difference between the density of basal bodies within these different parts of the apical domain. Situation is actually quite different when we look at the knockdown condition. So when we basically do the same quantification, so plot the number of basal bodies within these quarters, we see that there is a difference between the number of basal bodies within these different positions. So even though, as I said, the global density of basal bodies, so number of basal bodies per the entire size of the apical domain is not changed, the local density of basal bodies within the apical domain is actually changed. And in fact, we see this clamping of basal bodies. So here is quantified looking at the clamping factor. So again, we can see here if we kind of plot the number, or basically in that case is a variance of basically the density variance of the density of basal bodies per the mean density of basal bodies. And if you plot basically this parameter from the entire time points, we can see that there's a difference, meaning that there is indeed a clamping when it comes to clamping of basal bodies when it comes to the condition in which we knock down alpha-actin in 1. All right, so we see that there is indeed the local clamping, but we want to understand, what kind of drives this local clamping in case of this perturbation. So what we did in here, we start looking at which parameter is actually correlating well with this clamping factor. So what we did in here, we actually sort the quadrants according to the density of basal bodies. So on the left hand side we have low density of basal bodies, and towards the right we have the high density of basal bodies. So in that case, quadrant 1 is a low density of basal body, quadrant 4 is a high density of basal body, and we basically measure the intensity of actin within these quadrants. So what we see in the contrast situation, which was actually quite expected, that the intensity of actin, so mean intensity of actin within these different quadrants is actually the same throughout all of these different quadrants if you kind of look cumulatively through all of the time points. Situation is actually very different when you look now at mean intensity of actin within these quadrants upon knocking down alpha-actin in 1. So again, here we see on the left-hand side at the bottom, we now again sort the quadrants based on the density of basal bodies, and situation in here looks very different, meaning that the quadrant that has low density of basal bodies also have a low density or low intensity of actin within this quadrant. So this is, I said, this is again kind of looking at the cumulative numbers. So basically summing up all of the densities throughout the time points. But how does it actually look when we look in time? So again, if you look at the intensity of actin in time in these different quadrants, and please ignore this legend on top and just kind of look at the legend on the bottom, because the legend on the top is wrong. But anyway, so just please pay attention to the, let's say, quadrant 1, which is the pink color, which is basically is a quadrant. So again, here we have, okay, so just to be clear in here. So here again, we're looking at the intensity of actin at these different quadrants that we ordered according to the density of basal bodies. And here we actually plot this intensity of actin in time. So in the contrast situation, we actually see that there's no difference in the mean intensity of actin as we plotted in time. So it's kind of a flat line. But when we now look at the intensity of actin in time in the situation where we knock down alpha-actin 1, the situation is a little bit different. So here again, let's pay attention, let's say, to the quadrant 1, which is the quadrant where it has a lower number of basal bodies, so the pink color, and the quadrant 4 that has a higher number of basal bodies, so here in red, quadrant 4. So again, what is interesting here is that from the very beginning, these quadrants, have the low intensity of, so for example, the quadrant in which we have low density of basal bodies from the very beginning has also low intensity of actin, and the quadrant in which we have high density of basal bodies from the very beginning, they have the high intensity of actin. So this is quite interesting, because now if we, for example, look or quantify the number of the new basal bodies inserting into the apical domain, then the situation is a little bit different. So again, if you look, and again, just to be very clear, just that you understand. So now this plot is actually counting what we call new basal bodies. So basically, we look at the apical domain and we look at the signal as the basal body appears at the apical site, and we count it as a new basal body. Yeah. So now, if you look at these new basal bodies appearing at these different quadrants that have, in principle, maybe different number of basal bodies, different density of basal bodies. So in the control situation, actually, there's no difference, meaning that the new basal bodies are appearing at the same time with all of these quadrants. Yeah. But and this is actually the same for the situation where we knock down alpha-actinin 1, meaning that in the case of knocking down alpha-actinin 1 within the cell, the basal bodies are still kind of appearing in a way randomly between all of these four quadrants. So I think it's maybe a little bit confusing, so just let me repeat it again. So if I go back to this slide and look at the actin intensity within the knockdown condition, we see that there is a difference between the actin intensity in the quadrants that have low density of basal bodies. So basically, this quadrant has low intensity of actin. Nevertheless, the basal bodies are kind of appearing randomly with all of these quadrants. If this is unclear, just ask me again during the questions. But yeah, so that was interesting, because this actually prompted us to understanding how do you actually get these basal bodies to the apical site. And to answer this question, we actually look at the organization of actin within the volume of the cell. And what we actually realized was quite interesting. So now we're looking at the volume of the cell. At the top, we have the apex of the cell, and kind of looking towards the bottom using these cables. So these cables are very prominent. So these are actin cables in green, and in pink, you actually see basal bodies which are sitting on these cables. So this suggests that basal bodies are actually using these cables to insert into the apical site of the multiciliated cells progenitors. So with this in mind, we actually wanted to understand what's the function of these cables, or are actually the basal bodies using these cables to move from the basal position towards the apical site of the cell. So what we did in here, we basically removed the cortical layer of the cell, because this is the layer where you have a lot of actin, so therefore it would be very difficult to look at the cable, because here what we see is basically the maximum intensity projection of the cell. So looking at the cable is challenging in the first place, because these cables are, kind of a very thin structures which are positioned basically, vertically within the cell. So given the, very poor resolution in Z, it's very difficult to see them. So to see the dynamics of these cables, we actually remove the apical site of the multiciliated cells and make the maximum intensity projection. So what you can see here in green is the dynamics of these actin cables, and in pink is the dynamics of basal bodies. So again, you can see these very prominent actin cables, which are, you can maybe kind of think about it as a tree. So the roots coming towards the top of the tree. And you can see here, the maximum projections and in XY, and on the bottom, this is the basically XZ. So you can see here the resolution, of course, is very bad, but you can maybe more or less kind of get the intuition about the dynamics of these cables. And on the right-hand side, again, we can see the maximum projections looking at the basal body signal. And you can see here in the Z projection that these basal bodies are kind of moving from the bottom, and they kind of disappear, because this is the position where we remove the apical site of the cell. So now, to understand a little bit better what's the dynamics of these cables in the context of the movement of basal bodies, we actually quantify the intensity of actin in the different position of the cell. So we look at the intensity just underneath the expanding apical domain. So here we call it apical area. Then we're looking at the intensity of these actin cables outside of the apical area. And at the bottom, we look at the entire volume of the cell. So now if we look at this dynamics of this actin intensities, so again, looking at the apical area, we see that signal is initially increasing and then decreases. So this suggests that this cables actually forms and then kind of, dissociate, yeah? And then, however, when you look at the outside apical area region and the whole cell, the situation is a little bit different, because we see here that the intensity of actin is increasing and then starts basically plateau and it doesn't decrease. So this suggests that indeed there are these cables, and basal bodies are using these cables to move towards the top of the cell. And as basal bodies are, reach the top of the cell, so apex of the cell, the cables basically dissociate and get severed, or basically chopped into small pieces. So meaning that the intensity of the actin is not changed. Simply what we see is basically restructuring of the actin cables into the smaller position, or smaller length filaments. All right, so just the last point, or last kind of points about the dynamics of this basal bodies was, okay, where in the apical domain this basal bodies actually insert. So here we quantify the position of the basal bodies during the expansion of the apical domain. So what we see in here is that the basal bodies actually insert towards the perimeter, so towards the periphery of the cell. So they have a preference towards insertion towards the edge of the cell. And this is kind of interesting. So now, if I take all of this kind of information together, so what I showed you, we want to understand these three parameters, so three processes: how the basal bodies go towards the top of the cell, so towards apical side, how these basal bodies are get distributed and patterned within the apical domain. So taking everything together, this is kind of the model as we think. So it looks like that these cables are kind of growing from the bottom of the cell. The cable kind of hits the apical site towards the periphery. The cable gets attached within the apical site. Upon attachment to the apical site, the basal body gets inserted into the apical region of the cell, yeah? And then these basal bodies, what is also known from other studies, that basal bodies also generate also bring some sort of actin polymerizing proteins. So by bringing these actin polymerizing proteins, they also contribute to the expansion of the apical site. So as the apical domain expands, this generates additional size, additional space for another actin cable that again inserts towards the edge of the cell, and the basal body gets inserted, and basically the expansion continues. But now, if you now look at the dynamics of basal bodies within the apical site, what we see is that as basal bodies get inserted into the apical site, they get cross-linked within the actin network. Yeah. So initially, this cross-linking is very weak. So therefore, this is something what we observe, this transition from this sub-diffusive to the diffusive regime. Yeah. But as basal bodies get cross-linked within the actin network, we start seeing these pockets. So initially, these pockets actually can also diffuse by itself. So you can think about this entire structure, diffusing within the entire apical site. But as you kind of start growing more and more these pockets, so the apical domain gets kind of rigidified, meaning that these basal bodies or these pockets actually get stuck. So therefore, this is maybe, if you remember, that even though this entire pockets were behaving as a diffusing component, at some point, so at the end of the expansion of the apical domain, they kind of get more constrained towards the sub-diffusive regime. So now situation is a little bit different when it comes to the alpha-actin morpho, So when we knock down this crosslinker, so meaning that there's not so much of the changes when it comes to the insertion of these basal bodies towards the apical site. So we still see kind of a normal, the ratio between the number of basal bodies and the size of the apical domain. What is mostly happening is that there is no cross-linking of actin network. And because there's no cross-linking of actin network, there's no separation of basal bodies. Therefore, the basal bodies are getting together, getting clamped together. And this is the reason why you, at the end, even though you have normal kind of relationship between the size of the apical domain and the number of basal bodies, the basal bodies actually, they're getting clamped, and the local density of basal bodies is increasing. So I will stop in here. I would like to thank my, phenomenal group. And when it comes to this particular project, so that was entirely done by Raghavan Chegarayan, who is a postdoc in our lab. So there was also done in collaboration with Niels Bohr Institute. So here I would like to thank Jonas and also Paul Martin, and also Mandar from the Indian Institute of Technology in Bombay, who helped us with the theoretical model that I didn't talk about it today. So thank you very much for your attention.
Thank you so much. I think it was a very interesting talk. If people have questions, they can unmute themselves, or maybe just raise your hands and then unmute yourself. Maybe I can ask, I have a couple of questions. So the first question was, so in your alpha actin and knockdowns, why do you see a specific cluster? Why do you see clustering in just one quadrant specifically? And like, is the actin architecture in that quadrant different from the rest of the quadrants, or what makes that particular quadrant special?
Well, I think there's really nothing, let's say, special to this quadrant. So this is, let's say, we decided to divide the apical site into 4 parts, but, we can, of course, divide it into whatever number of parts. It was basically, in a way, for us, easier to, to get understanding about, is there any difference between different positions within the apical site. So therefore, in that case, we divide it into 4 parts, but we can also divide it in whatever, 2 parts, 8 parts, whatever. But what is different in there, and I actually haven't shown this stuff because it's still a work in progress. Actually, the organization, so the super resolution organization of the actin network is actually perturbed. So indeed, you don't have much of the actin, and if you have actin there is not really cross-linked. So this is, let's say, the major difference that at least we can quantify at this stage.
Okay. Yeah, I'll see if does anyone else have questions? Yes, okay. There's a question from Shimena. Does the clumping affect the waveforms or other behaviors of cilia repeating?
The clumping affects the waveforms or other behavior of the, okay. Yeah, to be honest, we haven't actually functionally validate what's the effect of the clamping of the, on the, let's say, the function of the multiciliated cells. I mean, I can only say that based on other works, when we observe clamping of this kind of type, there is indeed asynchronous beating between the cilia within the apical domain. So yeah, definitely there is a functional kind of repercussion of this clamping when it comes to the to the function, in that case beating of the multiciliated cells.
Yeah. Oh, very interesting. Is anyone else, if people have questions, they feel free to drop it in chat or just unmute yourself as well.
I can see the
I'm
question.
going to go if that's okay. I don't
Yeah.
know. So it's Jordan Raff here.
Yeah.
Okay. Sorry, I can't seem how to raise my hand. Something's updated.
That's fine. Just
Okay.
go.
Anyway, thanks. That was very interesting talk. I wondered about the generation of the actin cables. I was a little bit unclear. It sounded like you were suggesting that they were sort of basal body dependent. So do you think that they're all made actually when the basal bodies are below the surface and they then just reach up? Or, I was just a bit confused how you get that array of actin cables stretching down from the apical cortex.
Yeah, that's a good point. So I mean, the exact kind of timing of let's say appearance of these actin cables, I can't really tell you exactly, but I think that in a way, this is just my let's say gut feeling, that they are not really in a way linked to the basal bodies, meaning that we already observe in a way these actin cables before seeing proper kind of structure of basal bodies. So there must be, let's say, it could be that this could be 2, in a way, independent mechanisms, meaning that actin cables are in a way generated from elsewhere. I mean, we actually see these actin cables being in a way formed from the kind of on the membrane side. So it kind of reminds me kind of a stress fibers process from Johann Belesch, but could also very well be that somehow the formation of beta bodies is somehow happening at the same time as the actin cables are growing. But at least we see already actin cables let's say, quite earlier than basal body moving towards the superficial layer.
Okay, thanks. That sounds what I expect. Thanks a lot.
Cheers.
Yeah, just along those lines, I have another question is, so what actually causes them to sever? Because you mentioned that they sever after the basal bodies have moved up, but if they are assembled independent of basal body movement, why do you think there's severing of these actin fibers after?
Yeah, I mean, yeah, $1 million question. I think yes. I mean, short answer, I don't know. I would expect maybe that, as basal bodies maybe move on these actin cables, maybe there is some sort of change of the, whatever modification of these actin cables, or maybe there's another dynamics of the another protein that, that somehow goes, behind or at the bottom of the cable, and somehow the process is in a way synchronized, meaning that, you polymerize actin cables, basal bodies go towards the top. At the same time, there's a severing mechanism happening from the bottom or something like that. But yeah, this is just hand waving. I don't know.
Okay, there's another question on chat.
Yeah.
Is it known how the number of basal bodies is determined? And
Mhm.
can you manipulate the number, and do you predict that less basal bodies will impact on actin polymerization at the cortex?
Yeah, I mean, yeah, so I mean, the basal body itself, this is generated, let's say, generally speaking, in the structures called deuterosomes, so they're amplified from there. So is it known how the number of basal bodies is determined? Okay, so actually, I'm not sure is it known. I think I would say it's not really known. But what is interesting in our system looks like that the number of basal bodies is predetermined, meaning that if I okay, so meaning that when we start looking at the expansion of this apical domain and looking at the, let's say, number of basal bodies, it looks like that there is no, let's say, increase in number of basal bodies as the apical domain is expanding. But interestingly, I mean, there are papers showing, even these papers are kind of not looking at the dynamics, so they are kind of based on the fixed images. But in these papers, they can expand or make the apical domain bigger or smaller, and this is also somehow changing the ratio, basically the number of basal body inserted. So in a way, we can make the size of the apical domain bigger, and this also goes with having more number of basal body inserted. So this kind of linear relationship persists, but I don't know, how is this kind of, fits into the de novo generation of basal bodies. I would say that the number of basal bodies is fixed.
Okay.
My gut feel.
And then I think she also had another question. Can you manipulate the number and
Ah,
predict that less
yeah, sorry.
basal bodies will impact on actin polymerization of the cortex?
Yes, can you manipulate the number? Yes, yeah, you can indeed manipulate the number of basal bodies. It could be done. Do you predict that the less basal bodies will impact on actin polymerization? Yes, yeah, I think again it's interesting because, as I mentioned, to expand apical domain, you need to have, let's say, you need to have two things. You need to have polymerization, so basically the source of actin, and you need to have crosslinking to generate a pushing force in a way. So as I mentioned, basal bodies bring some of these, let's say, nucleators and crosslinkers. So therefore, I would expect, and actually it was also shown in some other papers, that if you perturb some of this, number of basal bodies, that the size of the apical domain would be smaller. Yeah.
Thank you. And then one more question from Dennis. Hi, Kuba, nice talk. Just a naive question. Are there any repulsive forces between the basal bodies that may play a role here? And are there any potential roles for microtubules here?
Yeah, cool. Well, I mean, kind of answering it from the perspective of the theoretical model that I haven't talked about today. In this theoretical model, we have to actually put some sort of repulsing force to model the process correctly. So kind of theoretically, there should be some sort of repulsing force. However, I would say that repulsion in that case is somehow driven by these pockets. So, the growing of this ring, this donut around the basal body, and this somehow generates the repulsion because before having this, let's say, very well-defined ring around the basal bodies, we actually can see basal bodies coming together, kind of, next to each other. Yeah, and around about the role of microtubules here. So yeah, I mean, good point. So this is, kind of a next step of this, kind of project or next projects. So, until now we have been predominantly looking at the dynamics of actin or actin network, but definitely there is a crosstalk between the microtubules and the actin network. We know that itself, this crosstalk is involved in the actually expansion of the apical domain, so it must be also involved into the patterning of basal bodies. We just haven't looked at these stages yet.
I think if there are no more questions, maybe we can conclude today's session. And I'd like to thank you once again for the wonderful talk. And just for the rest, we still have another talk left this term in 2 weeks' time, and you'll get all the information for that soon. Thank
Yes.
you.
Thank you very much.